Hi, my name is Sarah Morgan, and I am an undergraduate chemistry student at WVU. Over the course of 2 semesters, I have worked with Dr. Yahinu Agazi in the WVU Health Sciences Center on his research regarding potential cancer treatment methods, with a focus on breast cancer. The type of breast cancer we have focused on the most is triple-negative breast cancer, often referred to as TNBC. What separates TNBC from other breast cancers is the fact that it does not have progesterone and estrogen receptors and has normal HER2 protein function. The absence of these characteristics makes it significantly more difficult to treat, resulting in higher death rates among cancer patients. Fortunately, proteins associated with cell growth have proven to be useful in research, including β-Catenin. When beta-catenin presents dysregulated expression, it results in the formation of cancer cells. For our research, we compared the differences between beta-catenin positive and beta-catenin negative TNBC cells to determine the significance of this protein in TNBC and whether its silencing results in reduced cell growth and metastasis. The main goal of this is to find out if the concept of beta-catenin silencing would be used to treat TNBC, ultimately aiming to establish the grounds for future cancer drug research. To begin our experiment, mice with weakened immune systems were bred to create the parental, control, 4921, and 4991 groups. It was important for the mice to have weakened immune systems with the purpose of avoiding the rejection of the TNBC cells. The numbers 4921 and 4991 refer to the loci on DNA that have their RNA expression silenced, resulting in a silenced or reduced beta-catenin phenotype. This silencing was achieved using small hairpin RNA, or shRNA. These mice were then grafted with human TNBC cells. The cells used for this grafting were also placed in Eagle's medium for 72 hours and also in a separate soft agar assay for 10 days to observe under a microscope. We then took tail snips from the mice and extracted the DNA and proteins from the samples, analyzing them with immunoblotting for the presence of beta-catenin and other cell growth proteins. This not only confirmed that the cells used for the control were beta-catenin positive, but it also allows for future experiments in observing tumor growth and metastasis in living mice. Once the cells in the suspension had been incubated and observed over 72 hours, and 5 different cell counts were taken for every 24 hours, the relative percentage of these counts to the number of initial cells was used to create a bar graph comparing them. In Figure 1, the immunoblotting results indicated that the parental group used to breed further generations of mice along with the control group were positive for beta-catenin. The 4921 and 4991 groups were negative for this protein, as well as for cell growth proteins EGFR and SHP2. Beta-actin is positive for all 4 groups, since it is involved in cell structure, making it a suitable quality control for this process. Figures 2 through 4 features images taken at 24-hour increments of the cells suspended in Eagle's media. Control cells are depicted in Figure 2, 4921 cells are depicted in Figure 3, and 4991 cells are depicted in Figure 4. Initial observations show that at the 48-hour mark, both 4921 and 4991 cells experienced less growth than the control cells. However, by 72 hours, the 4921 cell growth suddenly spiked, indicating possible contamination of this sample. While the numbers of cells at this point exceeded those of the control group, there is an equal spread of 4921 cells across the suspension, while the control cells have formed larger clusters. This shows that control cells still exhibit increased tumorigenic properties in comparison to 4921 cells, despite the unexplained increase in growth. Figure 5 depicts a graph representing the relative percentage of the cell counts taken from the previous photos. The average cell count for the 5 images taken every 24 hours were represented as a percentage of the initial amounts in each sample. As can be seen in these images, both 4921 and 4991 experienced less cell growth up to the 48-hour mark, but 4921 ultimately spiked in cell count at 72 hours. Currently, there are no figures for the soft agar assay in tumor sphere experiments. This was because, over the course of these experiments, the cells did not experience adequate growth for producing usable data. This, coupled with the fact that these experiments can take several weeks to perform, made it so no images were ready for the symposium. However, the observations that were made for the limited cell growth that did occur correlated with the 2-dimensional cell suspension, with control cells showing increased tumor sphere formation. The results of this research implied that the silencing of beta-catenin is a factor in reduced TNBC cell growth. These findings suggest that once the oncogenic properties of TNBC in living mice are observed, Reduced tumor growth and metastasis will result from beta-catenin silencing. Such observations can then be applied to research and the development of drugs with the purpose of targeting TNBC and beta-catenin. While the silencing of beta-catenin appears to be related to reduced TNBC cell growth, it is still unclear if beta-catenin is the driving factor of TNBC prognosis. Even so, this knowledge is a key step in focusing research on cancer-related proteins and their use in treatment development. Such knowledge is especially important for cancers such as TNBC, whose absence of clear causation factors makes it much more difficult to utilize targeted treatment.